All right. Hello, everyone. I'm going to get started as people continue to join. Welcome to today's talk titled Reproducibility of Cardiac Mantarray Tissues in Context of Use. My name is Tiffany, and I will be helping guide us through this event, which is designed to be interactive. So please, if you could let us know in the chat where in the world you are tuning in from, that would be a wonderful start. This webinar will include a Q&A session after presentations, so please feel free to submit questions for our speakers throughout this event. For those of you who are unfamiliar, you can find the chat and Q&A functions at the bottom of your Zoom screen. Now it is my pleasure to introduce our speakers for this event, Dr. Christos Michas and Dr. Ayesha Arefin. Dr. Michas combines his background in engineering and biology to drive strategic planning and market alignment for Curi Bio's product portfolio. He oversees the deployment and continuous improvement of the Mantarray instrument and consumables, supports the productization of Curi Bio's emerging R&D technologies, and ensures a strong customer experience through close collaboration and relationship management. Dr. Arfin's expertise includes the development of organs on a chip and the evaluation of reproducibility in microengineered in vitro human organ platforms. At Curi Bio, she focuses on developing reproducible cardiac tissue models that mimic cardiac physiology for studying disease and assessing drug safety. And now, without further ado, I would like to hand over the presentation to our first speaker, Dr. Michas. Please begin whenever you are ready.
Absolutely. Thank you for the introduction, Tiffany. So Tiffany is absolutely correct. I'm Christos, product manager at Curi Bio, and co-presenting with Ayesha on how to generate reproducible, high-quality data from engineered cardiac tissues using the Mantarray platform. And so the webinar is split into two parts. The first part, I will give you an overview of Curi Bio's mission and technology. And in the second part, Ayesha will elaborate on principles and case studies of designing, interpreting, and generally making the most out of your cardiac experiments using the Mantarray platform. And so before we dive in, we would like to hear more about you. There's going to be a few questions popping up on your window in a few seconds. So choose the answers that feel right to you and scroll down to make sure you answer all three questions that are going to pop up. The first question is, what applications are you interested in? The second question, what is your affiliation? And the third one, how familiar are you with engineered tissue instrumentation? We'll give you a minute to be able to respond. That's great. Great. And we see there's a pretty good mix between all of you and what applications you're interested in. We have a good representation from all the different affiliations we let you choose. So this is great. And it's great because this presentation is tailored for all of you. So do not worry if you're in the minority. And so as we start the presentation, we're going to start with a bigger picture. So developing a single drug takes typically over 10 years and costs between $2 to $3 billion. And with that level of investment, you would expect that this system should work, but in reality, it doesn't. Over 90% of drugs that enter clinical trials ultimately fail. And that's not just a loss of time and resources. It is a sign of a deeper issue. These drugs didn't just suddenly fail in humans. They falsely passed preclinical testing. And the reason for that is because the models we rely on to predict safety and efficacy and even potency before human trials are fundamentally limited. Animal models often do not translate due to major biological and physiological differences, while molecular assays or 2D cell culture assays lack the complexity of real tissues and fail to capture meaningful human responses. What we're missing is a predictive model that reflects the ultimate outcome that we care about in the clinic, and that is functional change in the human body. In other words, we need human relevant functional models. And so let's consider how we can upgrade our models. 2D cultures have their place. Of course, they're simple, inexpensive, easy, and widely used, but they fall short in critical areas. They lack tissue architecture, cell-cell interactions, and essential functional outputs like contractile force in many cases. As a result, they struggle to accurately model complex human physiology. By transitioning to 3D models, we believe we can address many of these limitations. In a well-designed 3D format, we enable heterotypic cell-cell interactions within a more physiologically relevant tissue architecture that extends culture duration and, most importantly, in our opinion, captures functional outputs such as force generation. With the right architecture and readouts, 3D models can be predictive, more predictive, in fact, of clinical outcomes. That is our core belief at Curi Bio, and our mission is to deliver human relevant, functional, and translatable data as enabled by 3D in vitro models. On the top right, you can see real-world traces from our engineered muscle tissues. The top panel is the muscle tissue built in our Mantarray platform, and you can see that when you compare with the human and the mouse data right underneath, the patterns and signal characteristics are remarkably similar. This is what we mean when we're trying to close the prediction gap for clinical or beyond by bringing functionally relevant human-specific data from the clinic to your lab. And this core belief of ours is supported by a major shift observed on a regulatory level. With the passage of the FDA Modernization Act 2.0, animal testing is no longer required for drug approval. Also, the FDA in April announced plans to phase out animal models for monoclonal antibodies and select drug classes. Shortly after, the NIH as well echoed that direction with a renewed emphasis on human-specific research tools, particularly those leveraging iPSC-derived systems. And so the policies are changing, in my point. But even in practice, we at Curi Bio meet monthly with the FDA to support their efforts to modernize botulinum neurotoxin potency testing. The point I'm trying to get across is that regulators are speaking clearly. Functional, human-relevant data isn't just preferred, it's needed, and it will be the future. And this is a turning point, opening the door to human-relevant alternatives like Curi Bio's technology. In terms of our models, this is where we are and where we're headed next. In cardiovascular space, we're building models for congenital cardiomyopathy such as DMD, MYBPC3, in the context of hypertrophic and dilated cardiomyopathies. In the skeletal muscle space, we're advancing models for DMD and expanding into DM1, XLMTM, and atrophy-related conditions. And we're also addressing sarcopenia and aging-related muscle loss, supporting therapeutic screening to preserve muscle function. Do note that all these are areas where long-term force-based readouts are especially valuable. Also on the metabolic front, we're seeing strong interest in models for type 2 diabetes, GLP-1 pathways, and obesity-related drug development. And in the neuromuscular space, our Botox potency work shows how our platform and 3D in vitro models are already being used in regulatory contexts. Across all these areas, our goal remains the same: to provide human-relevant models that predict functional outcomes and accelerate therapeutic development and screening. These models are made possible by the technological infrastructure Curi Bio has uniquely pioneered, and it is a full-stack platform that combines human cells, specialized media, consumables, biosystems, instrumentation, analytics. It is all designed to translate biology into decision-ready data. And at the core are our Mantarray and Nautilite platforms, which deliver functional outputs. Mantarray, on one hand, measures contractile force in real time, and Nautilite maps calcium and voltage activity across 2D and 3D cultures. Stingray also adds programmable stimulation to support tissue conditioning and maturation. And of course, all these three platforms would be nothing without the underlying biology, correct biology. And that is why we support our systems with a range of iPSC-derived cells and specialized media for skeletal and cardiac tissues, enabling consistent 3D cultures. Finally, there is the Pulse Online interface, which is the unified dashboard for data analysis and brings everything together. And so within this, in the second half of this webinar, Ayesha will explain how the Mantarray tissues and the Mantarray system are using cardiac applications. And so I'd like to take a few minutes to remind you what the Mantarray exactly is. And so the core innovation or the core platform that we use is a Mantarray plate and the tissues, which you see on the top left. The Mantarray plate consists of 24 pairs of posts between which tissues are suspended, as you see here. Tissue creation is very simple. It uses a Mantarray lattice where the tissues are and a casting plate. The process is simple. All you have to do is place the Mantarray lattice into the casting plate, and two solutions are added sequentially, as you see in the bottom, to form a cell-laden hydrogel around each pair of posts. As the hydrogel gels and solidifies, the cells begin to compact, forming the fine tissue strip you see in the image on the top left. And this process, again, was designed for simplicity and reproducibility, and it can even be compared to something as simple as making popsicles, which is why with this process, you can achieve over 95% casting success rate. Once casting is complete, the lattice can be easily lifted out of the casting plate and transferred into a standard 24-well culture plate with media. And to change media later to maintain the tissues or introduce a new condition, all you have to do is just lift the lattice again, move it to a fresh plate, and that's it. No special tools required. The one thing that I didn't mention is that at the top right, you see, zoom into one of the wells and pair of posts. One of the posts, the black one, is rigid and solid, while the other one is translucent and flexible. And at the tip of the flexible post, you see this little dot that is a magnet. And so whenever the tissues contract, they're actually displacing the magnet. And so that's where the Mantarray instrument, the Mantarray system, comes in, which uses magnetic sensing to measure real-time contractile force from engineered muscle tissues. It's as simple as that. It doesn't require any dyes, any invasive probes, or any complicated imaging setups. In fact, it is small enough to fit in an incubator. It has the size of a shoebox. And so it also doesn't require changing your existing lab setup or even your workflow. And it is ideal for long-term tissue culture being in the incubator. Furthermore, the system provides electrical stimulation in each one of the 24 wells and tissues in the plate are individually addressable. So you can run multiple conditions in parallel, such as different long-term training protocols, but then dose response studies, etc. And so once you have made your tissues, all you have to do is take the plate, place it on the instrument, click a few buttons, and then you get in real time from the computer application, contractile waveforms like the one you see on the right for all the tissues in real time. And this is not just, and that's not the end of the process. This is not the end of the Curi workflow. From there on, this is where our process shines as a 0 to 1 workflow. Using the Pulse Online platform, each contraction is broken down into multiple metrics, and it's not just force. It is also time metrics like contraction and relaxation durations, the respective velocities, beating frequency, beating irregularity. And these detailed metrics give us a holistic insight into how a treatment or a disease state or any other condition affects muscle performance. And again, it's not just in strength, but also in how efficiently and consistently the tissue contracts and relaxes within time in a recording, but also across multiple recordings. And so from there on, it is really up to you to decide how you want to harness these metrics for your study. In this example of an AAV study here, we're using the muscle force to answer, the simple question, is my therapy working? So we have multiple conditions, multiple tissue conditions, each one with different contractions concentrations, excuse me, of the therapeutic observed over multiple days. And so the conditions, as you see, notably stratify to the point that one can observe where a threshold dose kicks in, what dose induces the highest force restoration, and at which point you might be running into functional toxicity or just not seeing an improvement. And that is exactly what makes Mantarray so useful. It captures the complexity of the biology and distills it into clear, quantitative decision-driving data. And so at this point, you might be wondering, so the Mantarray platform you told us lets us observe trends and differences between tissues. That's great. But how do I know the data is reliable? How do I design a study I can trust time and again? And what considerations go into building a robust experiment? And so these are great questions, and these are exactly the questions Ayesha will address in the second half of today's webinar, focusing on how to apply these principles in the context of cardiac tissue studies. And so as we now transition to the second segment of the presentation, a few questions will pop up in your window again. Please take a minute to respond. So the three questions are: what most influences your decision to adopt an in vitro platform? What do you see as limitations in tissue models? And would you be interested in a demo or pilot program with our platforms in your lab? Please take a minute to respond. Alrighty, quite a few results are coming in. Thank you very much, everyone, for responding. Again, we see a pretty well-distributed mix across most of the answers, which is very interesting, and that is great. Ayesha will respond quite a few of your concerns regarding reproducibility and standardization, as well as how to interpret data from our platform. Give you a few more seconds. Awesome. Thank you very much, everyone. And so with this, we are ready for the second half of the presentation. I will now pass on the presentation to Ayesha. Ayesha, whenever you're ready.
All right. Let's see. All right. Good morning, everyone. You can hear me, right? Making sure.
We can hear you, yes.
Yeah. Thank you, Christos. I'm Ayesha Arefin, the cardiac scientist here at Curi Bio. I was preparing to say good morning and a beautiful weather, but it's raining here in Seattle, so very reproducible. So I will cover how, as Christos already gave you an introduction on our Mantarray system, I will solely focus on the cardiac reproducibility in our platform. So the topics I'll be covering in this webinar is how we make stable functional tissue, what are different sources we identified as that cause variability. I will focus on 3 key metrics of contractility, which are the contraction regularity, relaxation time, and force. How this Mantarray tissue are reproducible when we are looking at a drug function and any cardiac physiology. And finally, what are different maturation strategies we are following to improve our consistency. So briefly, how we make the tissue, or we have cardiomyocytes, we have non-cardiomyocyte as cardiac fibroblast, and in a extracellular matrix, we have fibrinogen, matrigel, and thrombin. So once we cast the tissue, we culture them in growth media. After 4 days, we transition them to serum-free maturation maintenance media. Using the contractility platform, we record their contractile force over time, and that data allow us to understand how they're developing force, their beat rate, and their contraction and relaxation kinetics. So as I said, I will be focusing on 3 most important contractile parameters. The first one is the force. So when we have the tissue, it takes around 12 days to generate a stable force. And the outcome of this force, again, variable, and we tested it in 3 different conditions. So in our first row, you're seeing we have force data from multiple healthy iPSC donor. We have data from multiple differentiation batch from the same donor, and we have experiment to experiment variability. In the bottom half of the slide, we calculated the coefficient of variation of this force. What I am trying to get across here is we have established some QC parameters, and when we follow those QC parameters, this variability stays under 20%. And what does this 20% COV means? Think about you have a group of 12 tissue, and when we have a variability of 20%, that means with this group of tissue, you could confidently make a decision for if there is a change of force 14 to 18%. So that really gives you an idea how much samples do you need to actually look at the effect size. The second critical parameters I want to highlight is the beat irregularity. This is really important because if you can set up the range of irregularity you expect from your culture, that would lay the ground for any arrhythmogenic modeling that you would expect from your, say you're doing disease modeling, you are looking at a specific compound for safety or efficacy. So sometimes we wonder if this is a biological variability, technical noise, or is it actual differences that we are seeing. So again, when we are looking at different donors and from the same donor, but across different differentiation batch, we see the variability stays close to 20%. The different multiple differentiation batches, we see it a little higher. So I would add a little bit about the beat irregularity. Christos shows one of his last slides how you see those contractile waveform. So imagine one of your tissue has a 1 Hz beat rate. So I would expect a regular beat, and between those two beats, we will have a specific duration. So that is the interval. And when the tissue starts to have arrhythmic behavior, those interval changes. So this is not the only arrhythmic behavior we see in some of the disease model. We also see differences in the force. We also see differences in the deep twitch period. So looking at these 3 different irregularity in our tissue over time give us confidence when we are looking for a phenotype for a disease model. The third critical parameters is the relaxation time that directly give you the indication about the calcium handling of your tissue. So across these 3 different conditions that we tested, the relaxation time seems to be the more consistent parameters. So the variability ranges from 10 to 15%, and even the experiment, the variability even narrower. So that's this relaxation time, we expect them to be in the range of what we see in a physiologically relevant adult relaxation kinetics. And over time, we lay down the ground for this baseline. And when we are looking at any relaxation dysfunctionality, we look at if this cross the range that we usually see for this cell line. And if it does, definitely it gives us more insight about that disease. So now that I covered the 3 most important contractile parameters and how we are consistently reproducing them in our platform, I wanted to go over with the QC criteria that we are following that resulting these results. So I want to actually go through each of the steps. So first one is the cell viability. You could cast your tissue from a 2D pre-plated culture. You could cast your tissue directly from a frozen vial. So the difference is when we have pre-plated 2D culture, we get a viability above 80% and tissue are strong, beating by day 2. But when we have, we are thawing the cells and directly casting them, that actually have some revival time. And if the viability is less than 40%, we have noticed the contractile function is not as reproducible. So the first QC for us, if we are doing a direct thaw, it has to be above 40%. And if we're doing 2D, it's always giving us a very good result. And it's usually 70 to 80%. One of the other thing we routinely do is when we get a new cell line, we try to figure out what is the cell concentration per tissue give us the most stable contractile properties. Right now, for the mini tissue, depending on the cell line, we have a range of concentration starts from 350,000 cells to all the way to 500,000 cells. The reason it's important, some of the line, I would not say weaker, I would say different. when they have a lower force, and when we do that cell concentration with a lower concentration, we sometimes see they vary over the time. They never get to that stable position. So finding the concentration of cells in your tissue in the beginning of your program is really important. Third thing we look at is tissue compaction. So within usually 24 hours, we start to see compaction. And if the tissue doesn't compact well, that is usually a red flag. Your cell number might be off, or the cell might have died, or they're not producing enough force. So that's one of the way visually we could keep in mind, okay, this tissue did not compact well, let's keep an eye on them. Threshold force, 200 micronewton by day 14. So two different, or 2 points for this. First one is the signal to noise ratio. The 200 micronewton gives us the best results when we are looking at relaxation and contraction kinetics. And the second thing is, if the tissue is under 200 micronewton, if we are doing any therapeutic study, if we are looking at any risk assessment, the tissue lose more function, and it's really hard to understand the differences. So once the tissue reaches 200 micronewton by day 14, we know they're going to be a strong tissue, they will be stable. Other 3 parameters, we usually follow what is relevant to physiology. So adult beat rate, 1 hertz, and then we tend to see the irregularity stays under 0.1 seconds, and the beat period, which is an average of both contraction and relaxation time, under 650 millisecond. So once we have all this QC passed by some tissue, all the tissue, we go to the next phase of QC, which is evaluating this tissue for their cardiac function. So our favorite experiment is looking at their response to external calcium, and we do tend to look at their response to positive and negative inotrope. And currently, we are investigating their response to increasing force. Does, of course, mature tissue produce positive force frequency response? And then positive post-rest potentiation is giving an idea about their calcium reserve. And this is like an add-on experiment we do when we do the force frequency response experiment. So I will give you some example of how this stable tissue mimics physiologically relevant cardiac function. So the first one is the external calcium response. Again, we do have 2 different conditions. We looked at 3 different donors. We looked at same donor, same differentiation, but multiple experiment. So what should we expect when we do this experiment? For cardiomyocyte, their intrinsic property, when we increase the external extracellular calcium, they should have a positive force response to this calcium concentration. So between these 3 lines from 3 different donors, the response is very consistent, and when we look at their EC50, they're in a physiological range. And when we look at the same donor but 3 different experiments, 2 of them you would see they're almost identical. So that gives us a very good ground confidence that this tissue are consistently reproducing what they're supposed to do, the cardiac function. The second example I have is their response to isoproternal. So it's a classic cardiac drug, beta adrenergic agonist. So what we expect is when we expose the cardiac tissue to isoproternal, they should have a positive inotropic response, positive chronotropy respond, and then their relaxation time also would respond to these concentrations as well. So with the multiple iPSC donor, we do see the positive inotropic response, but it's the sensitivity is different. When we are looking at multiple differentiation batch and the experiments, different experiments, the responses are very close to each other. So again, they're proving when we follow this QC criteria and we have a very reproducible, stable tissue that physiologically mimicking all the cardiac function. So now I will transition our focus to how we are making more mature tissue and what our efforts are going on right now here at Curi. So positive force frequency relationship, a cardiac maturation hallmark. So very few instances are available right now in vitro technology that are achieving a positive force frequency relationship. Recently, we with our cardiac maturation media and the training protocol that we applied on this tissue resulted a positive force frequency. I'll walk you through how we are setting up this experiment, when we are testing them, and what we are observing. So we cast the tissue and we transition the tissue to maturation media on day 4. From day 4, we start training the tissue up to 4 Hz, which means day 4, tissue is spontaneously beating, and they are not up to 4 Hz just yet. So we ramp up the frequency and then try to see when tissue matches the highest frequency we're applying. So by day 10, I would say the tissue starts following this maximum electrical stimulation we're exposing them to. So we tested the positive FFR protocol for this tissue every 7 days to see when they produce the positive response. So it took them until day 18 to result a positive response. And it was consistent when we tested them again on day 22 and then day 28. One of the interesting things we see is when we start pacing the tissue, the force actually stayed in the minimum. You see the force graph is actually in the down. And the moment we stop pacing this tissue, it takes 24 to 48 hours for this tissue to go back up and match with the untrained tissue. So this is really interesting result, and we're super excited. And currently, we have reproduced it into only one cell line that we have in-house, and I'm working on reproducing this result in cells with other genetic background, and wanted to see if the same training protocol would apply for them as well. So I'm almost at the end of my presentation. I want to summarize what I have presented so far. So what I have focused on today is to get a reproducible and consistent tissue, we establish some QC criteria here at QC. And then when you follow this, we have been seeing the engineered heart tissue are reproducibly mimicking physiologically relevant cardiac function. And currently, we have achieved positive force frequency until 2.75 Hz, which is physiologically relevant. And we are working on reproducing this cardiac function in other cell line, and we're hoping this will reduce variability when we are setting up this tissue for downstream application. So with that, thank you for your attention, and I'm happy to answer if you have any question.
Thank you, Dr. Arefin and Dr. Mikas, for your presentations. We will now start the Q&A portion of this webinar. Please continue to send in questions. We will start with a few that we've already received. So one of the first questions we have is, how do you train your tissues to achieve a positive force frequency response? I guess, Ayesha, you can take this one.
Yeah, in the last slide, so it is a combination of the media and the electrical stimulation. So we cast the tissue, we transition them to maturation media, and we train the tissue to follow 4 Hz, which is our maximum training stimulation currently that we're working with. And once the tissue start following that 4 Hz, we continuously keep the stimulation going. So we change media every other day, and we after 18 days, that's when we do the force frequency test to see if they have enhanced maturity. Yeah.
Another question is, did you test to incorporate the Mantarray system in an automated setup? And if so, does this increase functional intra and interplate reproducibility? And a follow-up question to that is there a 96 well plate setup available?
So I can take this one. Thank you, Tiffany. So we have incorporated we have cast in the past with a robotic system. I can tell you that it's fully compatible. We do have numbers about reproducibility, and we will be sharing with them soon. So stay in touch. And I would say the same thing about a 96 well plate. This is TBT, so just stay with us, and soon you might hear from us.
And then another question is, how many tissues or wells do you on average need to generate reproducible, reliable data? So what's the N, typical N per experiment?
It depends on how your effect size. The smaller the effect size, you need higher N number. But from our experience with the variability we see, which is under 20%, seems like a 8 to 12 tissue per group would give you a 14% difference detection.
Another question, you mentioned that 200 micronewtons is needed to detect the contraction dynamics. What is your resolution of the system, and does that change with different stiffness of pillars?
I can take this one. So the system has enough resolution to detect contractions down to even 20 micronewtons, and mind you, this is about the 12x stiffness plate. If you would like to have statistical significance, so going back to what Ayesha said, it's really important to have signals that are maybe 10 or 12 or 20 times stronger than the baseline noise. And so therefore, we strongly recommend that you stick to the 200 micronewtons for the 12x post system, but you're free to go below if you can increase your sample size, for example. That, of course, is a 12x system, 12x stiffness system. For the 1x, because the stiffness is lower, the contractions are stronger. And mind you that the Mantarray system uses magnetic tracking. And so since the displacements are larger, then this doesn't apply. For the 1x system, I believe the equivalent force would be 20 micronewtons, something like 15 micronewtons, something along those lines.
Another question. I remember you had a strong DMD model in skeletal muscle. Do you have anything similar in cardiac?
I could take that. Yes, in our cardiac, we are currently working with two different DMD model. One is DMD exon 46 to 48 deletion. Another one is DMD E50.
And then another question for Dr. Arefin. Were experiments shown from cardiomyocytes from different iPSC lines performed in the same media?
Yes, they're all in the Curi media.
And follow-up question, how much does media matter? Can I use any media?
Media matters a lot. So we did actually do a comparison with currently there is two media in different publication people use for 3D cardiac tissue. One is DMEM based, another one is RPMI based. So what we have noticed, the RPMI based media have a very low calcium, which is not physiologically relevant, which actually results in very low force of the tissue. The compaction doesn't work as well. And when we are looking at cardiac function, we see very no to little function. The compared to high glucose media, the functions are very comparable, but one of the things we notice is with the high glucose, which is over time, the relaxation time actually changes and the tissue tends to crash. And we did not do more investigation on that as we were trying to put together physiologically relevant media where all the components in the media and the concentration are relevant to our physiology.
A bit more time for some questions, and I see a couple about cells. One is, are you going to introduce endothelial cells into your tissues? And what is the status of the cardiac fibroblasts that we use? Are they myoblasts or quiescent?
First, yeah, we use cardiac fibroblast is actually primary fibroblast from Lonza. Usually how we use it, we expand the fibroblast in-house, making sure they're in log phase, and then we freeze them down and we use the same passage for if we have few experiment from a study, we use the same passage to make sure we reduce any variability coming from this fibroblast. So that's number one. And two, endothelial cell. Yes, love to add endothelial cell in the tissue as well. It's a work in progress.
And I would also add to that the system is agnostic more or less to what cell types are added, as long as you can form a tissue that is contractile. So we just haven't gotten to it, but you're free to also experiment in your lab and try different cell types.
Here's another cell question. Do you see differences in cardiomyocytes derived from different clones or the same iPS line? For example, a wild type line and a mock CRISPR treated line, or is that something you've tested?
Yes, we have, actually I can give two examples. We have worked with a disease line and the isogenic wild type was corrected. We do see a big difference in their contractile properties, and unfortunately the wild type is the one that's the weaker line and unstable. And we have another differentiation batch was going, we were looking at two different clone, and it seems like from the same cell line, one of the clone has a like really high force and stable contractile properties over time compared to the other one. So yeah, we do see differences from clones to clone.
And here's a Mantarray question. Is the Mantarray compatible with optogenetic stimulation?
Yes, definitely is. Typically, we show data with our electrical stimulation lid, which you slide on top and into the plate. There is the equivalent for optical stimulation, we're calling laser ray. So you place it on top of the plate, and each one of the wells is illuminated. Again, it's individually addressable, and we have not necessarily used it for cardiac studies, which is what we're showing here, but for the neuromuscular junction projects that we have for botulinum neurotoxin, it has been essential for evaluating the transmission of the junctions.
And we have another cell question. Do you recommend using multiple iPSC donors for pharmacological testing, or is one donor line sufficient for pharmacology studies?
I would definitely recommend a multiple iPSC donor.
I guess a follow-up question on that. What is the range of forces produced by cardiomyocytes that you've seen, say on day 10, between different donors? What might the range look like?
I did show the first slide about this. So multiple donors. Right now, I think all the cell line we have worked with, there's two different category. One is like a stronger ones. By day 4, they're beating. They have contractile force crossing 400 micronewton and reaching to 1,000 by day 14. Where the little weaker line, they approach to 200 by, let's say, day 8 to 10, and they tend to just hang around that area the whole time we're doing experimentation with them.
Right. I think that wraps up our Q&A portion. If you think of any other questions that you might have about this presentation or any of Curi's products, please reach out to contact@Curi Bio. Thank you, Dr. Michas and Dr. Arefin, for your presentation and for all of your answers to these questions. And a big thank you to everyone who has participated in today's webinar. You will be receiving a follow-up email from us with access to recording of this event, as well as a link to access previous webinars on other various topics. And I hope that all of you found this webinar informative, and I hope you have a wonderful day. Thank you, everyone.
Thank you, everyone.